Okay, we can switch to the last session that is dedicated to the platform of Operando. And as in charge of the project, I will do a short introduction, in charge, in fact, with Pascal d'Oliveira previously, and now with Jean-Philippe Renaud. I will do a short presentation. It was already scooped by Rémi partially, but just on the role, on the objective, on the goal of this platform, and we will get few presentation to illustrate a few of the platform, and obviously you have the poster session that you can look and you can discuss with the people in charge of the platform. Just in this project, there's something in this PEPR. There's a E that corresponds to equipment, and that was a very nice opportunity, what is not very often the case, to have money, large amount of money to invest in platform that can be very important for your research. And we have tried to work on this to define a large platform that can survive after the PEPR if it's really answering the scientific question that you need. And we have identified a few challenges that are important for the community that are listed here, and the one on the bottom correspond to mainly the Operando. But I should mention that Operando and ultrafast are working exactly the same. It's just a limit that we have to define to write the project, but it's really a continuum between the two platforms. And if I try to describe quickly the platform, I can try to describe as a two-part. One is to characterize, the goal was to characterize the matters under illumination, and we have in fact included all the up-to-date or state-of-the-art technique that you can use in your research with STEM, EPR, NMR. All the technique that you need are introduced in this platform, and also some other that transform the matter under illumination that are also in this large hub platform. I think we have tried to do our best to select the platform. You see all the list of the platform of the Operando platform that are distributed in France. And I would like to mention also it's not only very up-to-date material, but it's also a competence and specialty of the people that can help you to develop the project with you. And that's very important also that we have selected the people that can contribute to your research by giving you the good advice. And the contact is Pierre Dufour that is on the first line. And just to be short, what we have achieved at the moment for this, it's at least we have used this money to start new equipment, updating equipment, and we have 15 operations that are realized already or in progress. That it was a big challenge to put this and start this. We have some human resource where we have hired 5 engineers that will help to set up the state-of-the-art equipment to do new experiment, new stuff that you need for your research. And we have access fees that are important for the platform, because the platform, as Rémi mentioned, it's free for the users. And also we can pay for your travel. That means do not hesitate to go to this platform. It's very important for your research, but it's also important for the future of this platform to prove that it's very important for this field of the research. And just quickly, the flow of to have access to this platform is the classical flow that you can find in the infrastructure with the criteria of access that are defined on the bottom. But I would like to mention also that the feasibility are are done by the people in charge of the platform, and that's very important that you can discuss with them if you have any doubt on what you can do, or do not hesitate also to potentially submit a first project, but also discuss with them to improve your project, and they are there to help you to create some new potentially experiment or new result, and that's really a network of stuff. Okay, it's open to everyone, not only the people that are included in Moonshot or project, as Rémi mentioned. And just to illustrate, obviously you can take time to have a look on the poster and discuss to the people in charge of the platform. That's very important. They can give you some advice and explain what they can do for you. Maybe you don't imagine what they can do at the moment. That means try to discuss. And we have selected two talk of people in charge of platform to show you maybe two platforms that you don't know so well. And also one talk of user to show you what you can expect to do in this type of platform. And I called the first speaker, Anthony Beauvois, for SOLEIL, and he will explain the photocatalytic reactor for X-ray spectroscopy.
Thank you. Good morning, everyone. So I am Anthony Beauvois from SOLEIL Synchrotron. So SOLEIL Synchrotron is itself a national platform with a large portfolio of techniques, including diffraction, scattering, spectroscopies from infrared to X-ray. But in the framework of the OPERANDO platform, we mainly focused on, not this one, sorry, this one. We mainly focus on X-ray spectroscopies and photocatalytic reactors. Our aim in the framework of this platform is to understand the lifespan of a catalyst under working condition. So to do that, we need the reaction condition. So it could be gases distribution system, of course, light sources for photocatalytic, and also eventually potentiostat for photoelectrocatalysis. As we want to progress operando measurement, we need also to measure the catalytic activity, so using, for instance, mass spectroscopy or gas chromatography to monitor the gases that are formed during the reaction. And finally, we need the X-ray spectroscopy to answer the question, what are the active sites, the active species of the catalyst? Why does the catalyst deactivate if it deactivates during the reaction? But the objective, the aim, was to develop reactors that allow to perform these three parts at the same time, meaning reaction condition, catalytic measurement, and X-ray spectroscopy measurement to perform real operando measurement. So the objective was to design specific cells or reactors that are suitable for the measurement, X-ray spectroscopy, to apply reaction condition, and to measure the catalytic activity, reproducing the same activity that we can observe in reactor at the lab. This is really important for us to be sure of what we are looking at. So in this framework, in this platform, we identified 4 specific beamlines that are represented here as a function of the energy range they cover and the time resolution they can provide. So we have Hermes beamline, Lucia, Samba, and Rock beamline. So as you can see on this graph, there is a good complementarity of these beamlines that allow to cover or to study specifically. This is the aim of X-ray spectroscopy, as was explained by the first speaker this morning. So with this complementary beamline, we can probe up to 82 elements of the periodic table, from the boron to the lanthanide elements. And in LUMA, we have there were an engineer that was employed at SOLEIL to develop specifically a cell for the Lucia beamline. The Lucia beamline works on soft, tender X-rays. So it's a primary vacuum environment, so it needs specific cell that allow to work under vacuum. So we developed these types of cell that allows to perform photoelectrochemical reaction. The cell here is in the vacuum vessel. So it has been validated from the chemistry point of view. So here we have the appearance of a photocurrent with titania, oxide titania. And very first results were done for the photodeposition of cobalt. And here we see that the cobalt is oxidizing with the movement, with the shift of the white line to the higher energy. So that's very first results that were done on the Lucia beamline. And now this cell is available for the community, for all the community who wants to perform such kind of experiment. But this is not the only cell. We have also cells, for instance, these types of cell on rock beamline. So rock is a hard X-ray. There is no the vacuum bottleneck on the beamline. So here is a gas phase cell for photocatalysis that is available and open to the community for photocatalytic measurement, and we can provide X-ray, UV visible sources. We have potentiostat. We have also the mass flow controller, the mass spectrometer, sorry, or the gas chromatography to follow the gases that are formed. And all this equipment is available for the LUMA community, and especially in September 2025, a project has been performed on Lucia beamline using this cell. I don't have the results now because the beam time was done just a few days ago, but just to tell you that there is some project that can be done. And for the SOLEIL, there is, of course, as it is already a national infrastructure, to have access to this infrastructure, you have to use the Sunset. This is the portal from SOLEIL. So this is a 2 pages proposal, and there is 2 calls per year with a deadline around mid-September and mid-February. So the proposal will be evaluated and accepted or not by SOLEIL peer review committee. And so I encourage every people who want to submit a proposal to contact me so that I can guide them to the good beamline, and we will help them in dimensioning the experiment for this specific for the proposal.
Okay. Maybe just a short question, if you have one. If not, you can discuss. There's a question.
Thank you. I just wanted to clarify for the proposal, it means that we need to specify the platform when we put the proposal, and then the beam line will be decided after the proposal is accepted, or is it just a part of the regular proposal?
No, it's part of the regular proposal. You should contact me, or if you already identify the beamline, you can contact directly the good beamline. But the best is to contact me so that I know the proposals that are submitted in the framework of LUMA. And something I didn't mention here, sorry, I forgot that. In the title of the proposal, when you submit it to the platform, in the title, start with LUMA. LUMA, sorry. And I don't know. And the title of the proposal so that we can put this in the LUMA framework, I would say.
Thank you.
Okay, we can continue with the, thanks again. And we can continue with the next talk that will be presented by 2 persons.
Yes, thank you. Okay, so we are presenting the PHOTOVIVO platform, which gathers optical methods for the characterization of biological tissues. And our research works are dedicated mainly to cancer diagnosis, but of course we can definitely adapt to any other application, which we already have, of course, as we'll show you. We have several optical and spectroscopic devices that we will not have time now to present, but feel free to come see the poster to talk about some equipment that may be useful for your works. We have unique and highly innovative devices, such as, for instance, the spectral radiometer that we may use in case you need to measure the biological safety of innovative devices that you may develop in your own laboratories. Like, for instance, we develop medical devices and we need to make sure that they are safe for the patients and for the physicians that use them in hospitals. In clinical trials. We also have a quality management system, which my colleague is in charge of. And today we are going to focus on 3 specific devices. One optical imaging device that is based on the LCOCT method, I will show you right after. One optical spectroscopic device that we developed and patented for autofluorescence and diffuse reflectance measurement. Those are applied to skin cancer diagnosis. And an optical bench for absorption, scattering, and anisotropy coefficients measurements that my colleague developed and that she will present now.
This device is an optical, the dual integrating spheres setup developed by our team to estimate the optical coefficient of biological samples as a function of the wavelength. So it's an absorption coefficient, the scattering coefficient, and anisotropy factor. The measurement bench consists of a supercontinuum laser emitting from 440 to 2000 nanometers with 2 integrating spheres and several detectors. To ensure an appropriate optical path length, the samples is about 1 millimeter thick and its diameter should be greater than 6 millimeters. So the sample is placed between the 2 integrating spheres, so here, and the laser beam passes through the sample and allows us to measure the light intensity received by the photodetectors and in order to obtain some information, 2 information, the transmittance and the reflectance. The transmittance, which is a part of the light scattered by the sample into the second sphere, and the reflectance, which is a part of the light reflected by the sample into the first sphere. The data is acquired with interface users that we developed also. To estimate optical coefficients, we use inverse problem-solving algorithm based on inverse add-in doubling method developed by Scott Prall. We have already performed some various types of biological samples, solid and liquid, like porcine blood, kidney stones, intralipid solutions, and live and dead yeast cells. So on the graph, we can see absorption coefficient spectra for different levels of oxygen saturation of hemoglobin, which we were able to measure with this setup.
I can propose also a focus on 2 other devices. So the first device that we have available for the community, just like all other equipments, is an imaging device based on LCOCT that was developed and patented by the French company based here in Paris, Damae Medical. It it allows a 3D imaging of skin tissues with a 1 micron spatial resolution. So like you can see, you can detect the cells directly in vivo on patients. And so we can use this during clinical trials. And the aim of a project that is currently finishing has been to evaluate the interest of combining this device by the Damei company with the spectroscopic device that you can see here in a surgery room at the hospital in Metz. That is based on spectroscopy to measure autofluorescence and diffuse reflectance, because we know that we have fluorophores inside the skin tissues that are modified with cancer development. So we base our diagnostic criteria on the measurements of such fluorescence. And here you can see the different spectra of different classes of skin cancers. Thank you for your attention.
Time for a question.
Yes, I think it's working. Thank you very much for the presentation. And do you think, it's a very naive question probably, especially your tool you mentioned with this double integration sphere, and so wouldn't it be useful for samples which are not always biological, not always for medical applications?
Definitely. We may characterize any type of sample, like we are part of the health topic of the PEPR program, but we may adjust to any type of application. So we were already contacted by people who do nothing related to health or biology. Yes,
okay,
of course.
Okay.
Yes, as long as it is transparent enough so the light can go through it.
Of course.
Can go through the sample and get some light absorbed, but detected by the second sphere, and some light that is backscattered if it's of interest.
Yes, thank you.
Thank you.
Okay, another question, or that's fine? Okay, let's switch to the last talk of the morning before the lunch. And where is the speaker? Ah, sorry. Okay, a user that will present his experience on one of the platforms. Okay.
Thank you.
So thank you for giving me this opportunity to talk about my experience. So I'm Christian, and I'm a doctoral candidate from IPCM in Sorbonne. And so how I'm working with Guillaume and Anna Proust, and we're studying basically photosensitization of polyoxometalates. And so we contacted and we partnered with LCBM in Grenoble to do some photoelectrochemical measurements on this system. So what we wanted to do is to see whether the photosensitization phenomenon that we were studying can be exploited and integrated into photoelectrochemical cells. So we contacted Muriel, Vincent, and Jennifer, and of the SOLEIL team, and to show us basically the know-how of preparing these systems, so preparing nickel oxide films and how to sensitize this film, so by grafting these dyes that we develop in our lab, and how to do some photoelectrochemical measurements, such as photocurrent measurements and also voltammetric measurements. And so this is just to show the setup, the kind of setup that they have in LCBM. So they have this in-house electrochemical setup that is also equipped with. They're also equipped with a light source that is capable of doing some automatic shutting on and off that allows us to perform chopped light photocurrent measurements, such as these figures that you see right here. And so these are the kind of data that that we got, and they also showed us how to quantify the quantity of dye into the nickel oxide film. So the idea why I did this is to learn how to do these things and to perform them in our facilities here in Sorbonne. And so in the end, it was very successful. So, well, this is just a last slide showing like one of the kind of data that I got. So it was just a short-term stay, but they were able to show us the things that we need. So here in our laboratory, we're able to carry out the same experiments and use the data for my project. So, yeah.
One question for this last talk. See, nobody. Everybody wants to switch to the lunch. Okay, we can thank all the speakers to this of this session. And enjoy this break to have lunch and also have a discussion with the posters, people in charge of the poster. Sorry.